Day 13. Side quests 0. Let's go. So Wave stopped yesterday, I think. 56.4. 7 questions left to go. But I also discovered interesting. How is that possible? I have a window on my OBS that doesn't exist. Dashboard, content, live. It is. So this is interesting.
Actually, by this time I knew that Mr. Puzzle was a great guy, but for the sake of this video's narrative, he must remain my nemesis.
Like, this is a very interesting game. I just discovered today, and it's remind me like the way how you optimize for sums. There is 3 metrics to optimize this game. You build different molecules basically with machinery and optimize efficiency or elegance. And those metrics are genius. I really love this video. I'm gonna give it thumbs up. Goodbye Galaxy. I wanna try playing it. But what's interesting, my OBS is stuck. Remove. I have to remove the whole scene now. macOS screen capture. No. Where's my display? Video capture device. No. Oh, maybe it was deprecated. Display capture. Wow. Okay. macOS main. And I think it's a BenQ, right? Yeah. Show cursor. Okay. We getting there. Okay. Here we go. I have to redo it completely. Today I want to dive deep into this. Like, this is, I mean, you can take a look and think about it like maybe busy or pretty cool, or what is it? Too complex? That was first reaction, right? But if you look at this picture in terms of how much energy and thinking was deposited to create this, that would be that would be through the charts. And I think like you don't usually see that metric, right? It's like infrared vision. Like when you see on something, you look, oh, you think it's normal, or thermal vision, right? You look at the house and like it's normal, and then like you see in one area of the house, like where the heater is the most red, right? So imagine this vision, which is not in a regular spectrum, but in a spectrum, and the metric is how much effort and thinking, the density of effort and thinking per square inch. That's the metric. Think about it. And not just think about it in square inches. Pun intended. Clip it. And many, many other unrelated but cool sounded words. Example of this deep thinking or density of thinking or density of energy, I can give example with New York City. I don't know if you've been or not in New York City, but when you get there, it's a strange feeling. It's like something that you feel something that you couldn't describe. And all those skyscrapers and so on, they are giving you a sense, and I couldn't figure out what that sense is, what it is. And when I try to apply that metric, density of energy that's been placed there by humans, by people, is actually insane. Through the charts, one more time. What does that mean? Just when you stay inside of it, and you know that every single building has been rebuilt at least 2 times, maybe 3 times, and every single brick has been placed by a human with a family or without a family who has a life, and then obviously more modern buildings are built by machines, but those machines all basically amount of actions, individual actions that has been made by human being in that particular island, it's through the roof. And now compare that city to Dubai, right? Also impressive. Also amazing skyscrapers, even bigger, even more beautiful, but it doesn't feel like New York. Why? Because the density is less. That's conclusion and examples that I can give, or conclusions that I came to, like thinking like why it feels different, like what's different about it. And so that's what it is. And this is just a beginning or kind of warm up statement before we dive into this beauty. Kind of give you the sense what we're gonna talk about it. Clip that and let's go. So, cognitive light cones. What is that? Let's figure out together. I mean, hello, obviously I know that what it is and kind of understanding it, but I want to go into the depths, into every single detail of this image, because I know every single word and detail, it has been thought through and picked from many, many other options that could be on that image, but that variation was picked for certain reason of deep thinking, first principle thinking, and many other principles of thinking applied to select exactly that individual element on a image to place into this Amazing. I would even say artwork. Yes, I will go that length and will call it a artwork. And art not in terms of like matching colors or looks pretty and so on. In terms of thinking process. The thinking, every time, if you drink every time, let's say thinking, you would be drunk already. But thought process here is so pretty. Let's dive. Before we dive, let's go into, let's check if we have a second version of it. Nobody can type this day and age, so I'm looking. Version. Cognitive Levin is a framework proposed by Michael Levin and collects to define the boundaries of self or cognitive agent based on its ability to pursue goals in space and time. So oh, interesting. ChatGPT image 2.0. So it's not, see, like maybe I was completely wrong. And we didn't put a lot of thinking to create this because ChatGPT created it. Ta-da! Roller coaster, right, of thinking. But let me check version 1 then. I want to find the original. This is the original. This is the podcast that's right here, right? Yeah, that's the original. So tick, dog. Right. This is version 1 and version 2. That's what we were actually looking. I'm wondering why there is a ChatGPT there. That's what's interesting about it. So time, it's a vertical one. Okay. 1 second. Time is a vertical one, space in the horizontal one. So here, now, future, past. Interesting. So it is like a cone, 2-dimension, where the space is actually in 2-dimension itself, but not really measured in 2 dimensions. No, it's not 2 dimensions. 1, 2. It's a space, and the length of the radius, that's what gives you a distance. Distance from organism on the right side. Body, microenvironment, territory, planet. Past, future, A, B, C. So the tick, a very small one. Dog. What's dog interesting about is that right away from the image you can see that it's dog care about the past way more than about the future. But you cannot see the radius, right? What it's thinking about, like a microenvironment, probably couple towns over. Not even, just a local town maybe. Human. Possible alien AI. Oh, okay. So possible alien AI Interesting. Possible alien, comma, AI. It doesn't think too much in the future or in the past, like, for example, a human, but it can think on the planetary level. And compound intelligent. That's even interesting. So colony is bigger than organism, and cell. That's how it's compounds. This is so cool. Let's go into this one now. See here, it's a little bit different. The dimensions flipped, so it's spread out to the center, right? I wonder for the reason of that. How life scales concern across space. For understanding minds, bodies, cells, swarm, and other goal-oriented, goal-directed system. It has a past, it can remember. It has a future, it can anticipate or predict, right? Intelligent is not a ladder. It is the size and shape of what a system can care about. Dog. Again, since the shape is pretty much the same, that's what I'm not really. Oh, here we go. This is better. Here now, space, past, bacterium. You don't even see. Cell, insect, dog, human, human civilization. All systems can be plotted in the same space. What I'm actually care about to create this, right? Is there a website for cognitive Lighthome? And what is measured as? Logarithm or not? Because everyone love leaderboards, right? So why cognitive Lighthome is not a leaderboard? And you can plug your CPU scores or your business or what you care about it, right? The humans itself, they could be measured. The measurements would be interesting, but still. Okay, so now the question is, we are building a website, right? We went to the biology. Right away jump into border between esoterics and like not conventional biology thinking theories. And now we're building a website. We went full circle. Obviously, we're gonna do it live. Obviously, we're gonna do it with AI agents. Yes, and it's gonna be very beautiful and satisfying. Now the question is, let's say I want to build a real-time visualization in a web browser where you can zoom in, zoom out into the 3D shapes and set my own scales. Like in a diagram, the vertical axis is a time. To the north, it's a future. To the south or down, up it's a future, down it's past. So our regular y axis, it's a time. The radius, it's what they care about. And we want to plot many organisms, as many as possible. And what's interesting, we also want to play around with subsets or within certain organism, how, let's say people, right, how they are related, not related, but how they represent it against each other. That would be interesting. Say hi to my beautiful wife. Yeah. She's the one who inspires me. That's a bad orange, sorry. And we were recording everything live, of course, obviously. So Galaxy apps, not related, but let's see how do they are related. Oh, it actually was muted. This is so cool. It muted my mic completely. A special place goes to the hive-minded and nested intelligence, like from the picture number 1. But the most important question, we would need a data. We need a data to fill that. Otherwise, it's useless, right? So how can we generate a data? Even if that data is synthetic and we can arbitrarily generate it based on some kind of formula that we can explain, that could be interesting. But question for you, do a deep research and give me a couple ideas. How can we score different organisms? And how can we generate a data for those scores? Let's go Opus 4.6 extended. We're doing extended and search. And yes, this is side quest number 1. Why? Because we still didn't answer those questions. Like day 1, we said we're gonna answer 50 questions. Day 13, we still answering them. But guess what? We learned so much during those side quests. That's, I think, more beneficial than just answering 50 questions. While it's digging, we actually gonna jump. Oh yeah, hold on. So bacterium, second 2 minutes memory, little prediction, local settings. See, like little. What is little? Little is adjective. I need numbers. Seconds 2 minutes. That's better. That's better. Tick, minutes, hours, memory. Interesting. Limited anticipation. Acts close to itself. Follows simple goals. Isolated cells and organism. Interesting. Short memory. Little prediction. Sense local chemistry. Simple regulation. So this is big question for me. Sense local chemistry. Is it only local chemistry, but can also listen, let's say, Wi-Fi? Why not? Why did we exclude it that cell can actually listen some electromagnetic waves? Who said no? I mean, maybe someone already said no, and I'm just unaware. Totally possible. But I'm gonna question that over and over. The goal space. The goal space, so all this space where those goals could be set and achieved. So what's interesting also, by setting those goals and achieve them, they get a reward. And the farther goal from the center, the better reward is. So if you want to optimize reward, that's interesting. Like, are we going for the long-term goals or short-term goals? Like, are we TikTok scrolling or we setting a destiny life level meaning and goals? And I think that's where Maslow pyramid comes in mind, right? As well. In the beginning, we set in a short one, but when they satisfied, we want to expand. Large cons, planning, abstraction, distance goal, hours they monitor plans and predicts, navigates space. Yeah, what I don't like about these cons is that they look the same size. And obviously I don't know how to show them differently, only in 3D and interactive. Design goal, fast communication, models at scale, act across networks. Okay, this is pretty clear, like pretty clear. Individual with small cons. Con. Bigger cons emerge with parts become a collective. So they become a collective. Each cell has a tiny word of concern. Gap junctions and bioelectric signals let cells share information, synchronize, and coordinate. So that's another thing. It's like it was said that's only chemical signal listen. But in organism, right, in a colonies or aggregation of the individual cells, now they have bioelectric signals. And then large collective cell. A large collective cell. The group can now detect model and regulate more, becoming an organism with larger goals: maintain autonomy, heal, reproduce. Makes sense so far, right? Bioelectric communication helps cells become a collective self. Shared signaling expands what the group can detect, regulate, and preserve. So right away comes in mind like we are individual human beings. And when we going into communication and collective self, right? What help us? Internet, right? We have ability to talk to each other, but now can we talk in a distance? Internet, information. But what then? What higher goal could be set, achieved? And what communication level do we need to communicate with different intelligence? Not just communicate in order to collaborate. And that's what's most interesting, is that maybe we already are through the tiny interface, thin interfaces. We communicate the way how we think, because we think like in visuals, words, and they are communicate through. We don't know how they think, but we can communicate through those tiny interfaces, and that's what Michael Levin with his team actually researching and mapping those interfaces. Through those interfaces, they trying to map a platonic space as some space where the other intelligence exists that's trying to communicate with us. And this is fascinating, to say at least.
Over the next few years, I would keep setting records, mostly in the journal puzzles. My mantra was cheaper and slower. I found that many of the existing sum records were tuned a little too fast.
Amazing. I was saying all that without you hearing me. And that's interesting. This applies to all life. Let me see in this research. Now I have enough to give you a solid synthesis. Here's what I found across the research, and that's the side quest number 2. We building the website. Different flavor and grounded in real science. The core problem, Levin himself acknowledged, there is no established quantitative method yet. In his own words on X, the theory of how to draw cognitive light cones in various spaces is very much under development. So anything you build is generally novel territory, which is both the challenge and the opportunity. The cognitive light cone has 2 axes that need scoring. Temporal, yes. Spatial axis, correct. Approach 1, the composite proxy model, data-rich, formula-driven. This uses real biological measurements as proxy inputs that combines them into synthetic inputs. Working memory duration E. Equally adaptive memory is roughly 1 second. Squirrel monkey. episodic memory depth, predictive horizon, spatial score inputs, home range as goal space proxy, cortical neuron counts. I don't know about this. Inception counted. I flawed but still useful as one axis. Humans and dolphins for chimps and dog. Encephalization quanta. I don't even know what's that. Data generation. You seed maybe 40 or 60 species from real data, then interpolate the rest using allometric scaling law. Brain mass, body mass, home range, body. But this is only for the brain. Our organisms, right? How are we gonna plug intelligence that's not biological? The allometric relationship lets you fill in hundreds of species from just body mass. Okay. Approach to the search efficiency model, K-score, mathematically elegant. This comes from the brand new 25 synthesis, cognition all the way down to point Y, which formalize intelligence as search efficiency K. Okay. K measures how many orders of magnitude more efficient an agent is at navigating problem space compared to a blind random walk. The paper gives concrete K values. I love this. That's what we're gonna do. Because that's we can simulate. We can take a cellular atomica game, set it at random, and then give some kind of brain or intelligence, like simple ML, and compare them to. Leave that. How you use it for the light cone. The K-scores map naturally to the cone radius at any given temporal slice. A system with higher K can pursue goals across wider spatial domain because of the decayed function. For nested hive intelligence, this is where it gets interesting. The paper notes that collective amplify K. A bee swarm acts as a single reinforcement learning agent. There's 2024 ARXIV paper literally titled The Hive Mind is a Single Reinforcement Learning Agent. So for nested system, you'd model when n is group size and coupling coefficient represents the cognitive glue. By electricity phenomenon, Stigmery language. Tightly coupled system, molecular organism gaps, junction connection tissue have coupling near one loosely. Data generation. Unit K estimates for anchor species. The paper gives a few, then it drives the rest from metabolic scaling, behavior complexity classification, the synthetics general formula. Interesting. Where behavior complexity, the chemotaxis only one learned associated tool. Approach 3 is the multidimensional goal space model. Most faithful to Levin. Explicitly says the light cone is about goal space, not sensory motor space. A human doesn't just have a large spiral reach. A human can care about the welfare of every individual and in the Amazon. That goal has a spatial scale of thousands of kilometers, but the zero physical reach. This approach scores organism on Levin's own framework components. Homeostatic complexity. How many set points does the system maintain? A bacterium maintains a handful. pH osmolarity nutrition gradient. A human cell maintains hundreds. A human body maintains thousands. Score number of independent homeostatic loops. First of all, thank you for those 12 days. Without them, I had no idea what this means. And now I understand. So all those loops and systems that self-regulate inside of us, we can count them. Goal abstraction level. Can the system only pursue physiological goal, food, temperature, or behavior goals? Plasticity. Can the system reach the same goal from a different starting state? That's very important. Persistent. Does the system keep working towards a goal despite perturbation? How large perturbation can it recover from? The cone shape formula. This is very good. Practical recommended for you build. To go with approach 1 as foundation, K-score from approach 2. Approach yes, for data generation yes, 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 yes. Want me to start building the dataset file and the visualization? No, we'll build it. We'll do that in a Claude code, CLI, but I want you to do a deep research in terms of how you're gonna visualize that with JavaScript and CSS in browser. And what I want you to do to generate me a prompt that I will set to the new agent to do a deep research that will search thousands different CodePen solution and pick which one we can apply or could be useful for this visualization. And I believe it's a CodePen, right? Oh. Yeah, this one. Let's see. Maybe even one question. What is the difference between antibiotic and antibiotics and antiviral? This is very interesting question. And as far as I remember, antibiotics and antiviral, the medicine that represent a group of antibiotics or medicines that represent a group of antiviral are different because for antibiotics, I believe that medicine aim to break a membrane of the bacteria, so in order to kill it. But for antiviral one, it doesn't aim to kill membrane because majority viruses doesn't even have membrane. They have protected SAG. So I don't remember. Yeah, it was. I don't remember. We touched that somewhere in the last 12 days. But antiviral, probably to slow down replication. Oh no, to antiviral, the viruses actually bind to the proteins on a on a cell's membrane in order to get inside and replicate themselves. Use in normal cell machinery to replicate themselves. So the antiviral one probably blocking those receptors, so the virus couldn't get inside and macrophages just eat it. Then search category specific search queries speed delivery format priority. The harder problem are flagged first. Logarithmic multi-scale zoom in 3D in and order independent transparency for nested cons. Those are the two things where you either find a good existing solution or you'll be written custom shaders. Everything else has well trodden path. I actually want to handle logarithm on a backend, and for front end, I just want to give a regular 3D space. But it will represent logarithmic numbers, but we shouldn't do it on the front end. We can convert them on a backend from a logarithmic scale to regular scale and just give you like coordinates. Does it make sense what I'm saying? So the whole solution doesn't have to be on the front end. We can keep the data on the backend, then have proxy API that will convert. Because maybe we'll use logarithmic today, maybe we'll use some different formula. So we definitely need a proxy that will take a raw number from database and applying different formula to represent it differently in 2D space for the browser that will be used for 3D visualization. Strong reasoning. You derived plausible mechanisms from first principle. Let me mark it up. Antibiotics and antivirals are fundamentally different. Correct. This is the key insight. You can use antibiotics on viruses, and antiviral don't work on bacteria. Antibiotic breaks the membrane of bacteria. Partially right. Some antibiotics do target the bacterium membrane, but the most common mechanism is different. Penicillin are related antibiotics target peptidoglycan, the material in bacterial cell wall called 2Q. They prevent bacteria from building or maintaining the cell wall. Without the wall, the bacterium swells from osmotic pressure, calling to question 21 osmosis. Osmosis, I don't remember that. Osmosis is probably osmosis. Spontaneous movement of water solvent across the membrane from the region or lower solution. Okay, osmosis. That's the water goes from a less dense to more dense, I guess. Not a dense, but a mineral one. And bursting. Other antibiotics works differently. Some blocks bacteria ribosomes from making proteins, some block DNA replication, some disturb metabolism pathways. A common trait antibiotic explore features that bacteria cells have, but human cells don't. The whole different ribosome structure, so they kill bacteria without killing you. Love it. I need a bathroom break. We're back. Viruses don't have a membrane, they have protective coat. Mostly correct. Viruses have a protein coat. I mean, yeah, capsid. Some also have a lipid envelope stolen from the host cell. Whoa! So they dress like a wolf in a sheep. But the core structure is the protein shell around genetic material, much simpler than a cell. Antivirus probably slow down replication, correct. This is exactly the main strategy. Block receptors so viruses can get inside. This is one real mechanism, but not the most common. Most antiviruses work after the virus is already inside the cell. Block virus enzyme needed for replication. Block viral enzyme needed for replication. HIV, all right. Drugs block reverse transcriptase, so viral RNA can become DNA. Totally makes sense. Like, this is so interesting. Now I understand what they're saying. And it's been only like 12 days, 13 days. So viral DNA can splice into your chromosome or block protease. So new viral particles can mature. Each drug targets a specific step in the viral life cycle. Block viral release. Tamiflu for influenza prevents new virus particles from escaping the infected cell. They are made but trapped. But why a cell is not bursting? Block entry, what you describe, Maravirus and HIV drug blocks the CCRP receptor on T cells so HIV can't dock. On T cell? This is a real, just not the most common approach. Why antiviral and are harder than antibiotics? Bacteria are cell with their own machinery, lots of unique targets to attack. Viruses use your cell machinery, attacking their replication process, risk damaging your own cell. It's like trying to stop a hijacker who is flying your plane. You can't just shoot down the plane. Every antiviral has to find something the virus does that your cell don't. And those targets are few. 6. Interesting. Fundamental reasonable mechanism, yeah. What is the ribosome and what does it do? Finally, this is a good question. I think a ribosome, it's a big molecular that's, or organella even so, or molecular probably, that's actually, it's a machine that's read RNA and build protein from amino acids. I might be completely wrong about that, but let's go. Correct, it's technically both. A massive molecular complex made of RNA and protein large enough to be considered an organelle. It's one of the few structures in the cell made partly of RNA, ribosomal RNA, or rRNA, rather than being purely protein. Reads RNA, correct. Specifically mRNA, building proteins from amino acids, correct. That's the entire job. The ribosome is a translation machine from the central dogma, DNA, RNA, protein. A ribosome does this last step. It slides along the mRNA, reads each codon, 3 nucleotides, and for each codon, a small adapter molecule called tRNA, transfer RNA, brings the matching amino acids. The ribosome links them together one by one into growing chain. When it hits a stop codon, it releases the finished protein. A few details worth knowing. Ribosomes are fast. They add about 15-20 amino acids per second. This is crazy. Cells have millions of them. A single active cell might have 10 millions. No way, no way, 10 millions ribosome? So every second we can assemble 20, no, not 20, from 150 to 20 to 200 millions amino acids. The most abundant molecular machine in any cell. Do they all identical? They all identical. Multiple ribosomes can read the same mRNA simultaneously, each producing a copy of the same protein. A string of ribosome on one mRNA is called polysome. It's like a production line. Free ribosomes float in the cytoplasm and make proteins that stay inside the cell. Bound ribosomes are attached to the endoplasmic reticulum, the RAF RAF because it's studded with ribosomes, and make proteins distant for export or for the membrane. This is my this is my first legit point, a first time. That's a good way to finish a stream for today. My first point, clip that. Day 13, 2 side tracks, first solid answers, even so I didn't know the answer before I started the study. So what's that? 44 questions gave me enough base to finally get it. 44. 13 days. 100 side tracks. Love this. Plasmid and why do scientists care about them? No idea. No idea. Cell speaking combinations. Oh, I remember that was that. That makes perfect sense. Damn. Called it. Clean separations. The front end become a dumb render. The backend owns the math. Swap the projection formula. The front end doesn't even know it. Yes, let me update the prompt. Now let me update category 3 since the now update the now update the no. Oh, architecture site and raw data store, Go proxy API, JSON browser render. Category 3 reframe entirely. It's not longer about how to logarithmic exactly. That's what I'm talking about. About how to zoom. Like, I can read technical documentation way faster than biological. Like, you see this? I'm already. That's probably practice, right? The search query shifted from log scale to the category 10 now covers API driven scenes update. Yes, please. New deliver 6 amino strategy. Priority order reshifted. Love it. Let's go. What it is? Probably a cool, amazing prompt. Deep research prompts. Yeah. Okay. Yeah. Interesting. Makes sense. AI. That's AI slap. See how fast I'm reviewing this? We need this. Do we? D33D. Better name. Category inspiring magic visualization sequence. Very interesting. Deliverable for Mart. I'm so excited. Let's start it. So we're going into Add project, biology, click here, doing research. I wanna run an Opus 4.6. I'm not fully trusting the 4.7 yet. Let's go, guys. Today been a blast. Thank you so much, and see you next time.